Okay, thank you. I am trying now to share my screen. Does everybody see a desktop with BioNumerics?
I do, yep.
Okay, good. So we've talked a lot about BioNumerics over the last number of years, and the BioNumerics versioning in terms of the tools that have been available have been changing, as most of you know. So if you've attended a BioNumerics workshop in version 7 at some point over the last few years, there's probably been a lot of changes since then. And for most of you, you're probably not using BioNumerics, with exception to maybe a few of you that are Listeria pilot laboratories and have some familiarity with the version in that. But we have also enhanced a lot of tools there as well. So instead of going through a PowerPoint sort of showcasing, all these things in sort of a flat format, I thought it might be a little more beneficial for you guys to actually see it in action. So hopefully this BioNumerics demonstration will go off without any technical hitches, but that's always a threat when you're doing something live. But to start off, I do have a couple PowerPoint slides just to orient you all on what we're going to be talking about. And one of the biggest things that none of you have used yet is our reference ID database. So this is going to be a new component to the BioNumerics platform once it's released to the States for whole genome sequencing. Um, this is sort of a schematic of the workflow, how we see it at least in the public health laboratory when you are using BioNumerics to analyze your data. Most of you have seen this in other presentations already, but what's going to be different here is, of course, we're going to see this all sort of in live action. So all of you should be able to sequence isolates at this point. The big difference between sequencing isolates prior to using version 7 versus post everyone moving to version 7 BioNumerics is the file naming format. So before you would email PulseNet at cdc.gov and request a WGS ID, we would provide you back that ID, and then you would name your sequencing files on the MySeq machine with that ID so that your file naming format, when it comes out, has the PNUSA number with your lab ID, the machine name, and a date, along with the other additional stuff that MySeq program adds to the file name for your FASTA files. Moving forward, though, you'll be able to use your just PulseNet key name in place of the PNUSA number. So that's the biggest difference there. The next thing is saving your data. Nothing will be different there. You can save your data on BaseSpace. You can save your data locally. This is the sequence data that comes off of the MySeq machine. And then the new part will be your reference ID database. This will be a locally created database on your computers that you will be able to link all the data that you have just run on the MySeq machine, submit it to the calculation engine at CDC for de novo assembly, as well as species contamination detection and ANI identity. So you'll be able to identify your genus and species there. After that, there's going to be some quality metrics you can verify to make sure that the sequence quality is passing, and then you can export those entries from that database. After that point, you can go into your organism-specific databases, open up that database, import the entries that were exported from your reference ID database, add in any demographic information. You can either key it in the computer there, or you can import that information. You will then submit again to the calculation engine, this time for allele calls and your genotyping results. There, you will also have another opportunity to verify that the final quality is passing, at which point you would upload that sequence data to the PulseNet national databases. Upon that upload, a WGS ID is manually assigned to your data or to your upload, and then you can upload it to NCBI so that it's de-identified there. So one question we've been getting a lot is, what is identified in the reference ID database? Is it anything and everything? Is it just PulseNet organisms? So we do have a chart here, what ANI identifies. So it is specific for most of PulseNet use. So if you were to bring in any of these organisms into your reference ID database, we would expect ANI to be able to identify all these different species of Campylobacter, Escherichia, Listeria, Salmonella, and Vibrio. Now, our WGMLST schemes were not built to provide basically annotated allele calls for all these species. For instance, with Listeria, the scheme was built to focus on Listeria monocytogenes. So if you imported, for instance, a Listeria innocua into your Listeria database and submit it to the calculation engine for allele calls, you may not get very many calls and you won't get good quality data. So everything you see with an asterisk are things that you could identify in reference ID, but you wouldn't necessarily import or export those to import into your organism-specific database. If you did want to do further analysis on those, you could do a SNP analysis, and BioNumerics does have that capability as well. And we'll come back to this in a moment, but we can go ahead now and start with the demo. So you'll have your BioNumerics databases here. You'll have your databases created. I have, for demonstration purposes, an Escherichia database, as well as a reference ID database. So what I'm going to do is I'm just going to open by double-clicking on my reference ID database. And the database opens, and I could see some entries here already. If I got a new run and I wanted to import entries to do my reference ID work, so at this point, the sequence data came off the MySeq. I'm ready to import it into my reference ID database to see what I have. So what you do is you go to your import data into the database icon, and then you import sequence read sets data as a link. So we're going to link to the files. You can link, again, to them from BaseSpace, a local file store, in CBI even. But in this case, I have my files linked locally, so I'm going to go navigate to where that location is by browsing. And I just have these on my desktop. So these are the files that just came off the MySeq that I'm ready to import. And for the sake of time, I'm just going to grab a couple. Otherwise, it might take a minute. So I'm going to select these two, read 1, read 2 of my key number 18 Georgia 1060. That populates this. I can import as much as I wanted to here, but I'm just doing these two for, like I said, the sake of time. I then will need to decide how I'm going to import this information. So we've created an import template. So as you know, let me see if I can actually edit this to show you. I want to import this data into the database based on its key name. So if you look at the file name, you'll notice that the file name is pretty long. It's got the key name associated with it, but it also has the Georgia part, the machine name, the date, and then all this other information. So when you set up your template initially to do importing, what I want the BioNumerics to do is parse off the key name so that it only imports this part of this file name. So by doing that, you just edit the parsing, and then you go to data parsing string, and then you decide which data you want to keep, and then at what point you want to parse. So I wanted to say I want to keep everything up into the first dash. So that's what that does. And if I hit preview, it shows that I'm only left with the key, which is the key that I'm using for the PulseNet database. So that's fine. And then you just choose next, and then you select your linking variable. In this case, it's the key, and then finish. So once you've created that template's created always. So as long as your file naming format doesn't change, it'll always parse that same way. And then you get to the screen where you're either creating entries or updating entries. So if you already have entries that you're linking data to existing in your database, of course, you should be updating. If you are creating entries, of course, it should say create. So it's always a good time to double check here before you just get in the habit of clicking next, 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 that you are doing what you think you're doing and creating an entry or updating entries. If something says creating when it should be updating, then you may have parsed something incorrectly. You can go back and edit that. Anyway, though, we are creating a new entry, so we just hit next, and then we hit finish. And then it tells us here that this entry has been created, and now we are ready to submit it to the calculation engine for a contamination check and for a de novo assembly. We also are going to do ANI in here, but we have to do ANI after the de novo assembly is finished because that's what it relies on in order to determine the species and genus. So I'll just uncheck ANI to start and then choose OK. Now, if you're linking files locally, BioNumerics has what they call the CE store. This is a program that basically links your files and then uploads them to the calculation engine. So that's what this information box is telling me, and that's OK. And this is what the CE store looks like. You should see this window pop up, and you should see successful progress of these two files being uploaded to the calculation engine. So you want to leave this box open while the uploading is occurring. And once it finishes at 100%, like it just has, you can close this box. At which point, you see that the entries have successfully uploaded to the jobs overview window. This is where you see the status of the job in the calculation engine. So what you can do is you can hit refresh, and it'll change statuses as the statuses change. So you would see queued, waiting queued, then you will see running, and at some point you will see finished. So for the sake of time, running a de novo assembly through the calculation engine can take anywhere between 30 minutes and an hour. So we obviously don't have that kind of time to wait for this job to finish. So I went ahead and already did that for a few entries. This is just what it looks like though. It says finished, and when it says finished, you can just highlight them, and then you retrieve these jobs from the calculation engine by hitting this gear with the arrow coming out of it. So for these jobs, I've already submitted the de novo assembly, and then I submitted them again for the ANI. So what we're retrieving now is actually the ANI results, because the de novo results have already been retrieved. And so if that's done successfully, you'll see that 5 results were imported for 5 entries. And then these guys are still waiting queued. So we can close this window. This window does not have to be remaining, or does not have to remain opened. So we can close that while those jobs are being performed. And we can see now that for these 5 that were done earlier, I did get the de novo assembly and the ANI results, and I have a Listeria monocytogenes, I have a Salmonella enterica, a Campylobacter jejuni, a Vibrio cholera, and an Escherichia coli. So some of the things you can do is you can go here to your quality experiment, number 4, and you can click that dot. This will pull up the different quality metrics that were captured through this analysis, and also if there was any contamination. So if you scroll down, you see contamination, secondary coverage. You're really looking for a number here that says 0. So that means that this is not contaminated, so we're good there. Some other metrics, there's a lot of metrics here, but we don't necessarily look at all of these in order to assess the actual overall quality. We look at a few specific metrics so far, and some of those would be the length. If we're dealing with Listeria monocytogenes, we're expecting anywhere between 2.8 to 3.1 million base pairs. This length falls within that line, so we are good there. Number of contigs typically are less than 100 for Listeria, so we've got 19, that's really good. We have average read quality. This number should be over 30, and it is, so we're happy with that. So those are a few of the metrics that we look at in order to determine whether or not this should be moved on to the organism-specific database. Once we get there, there are some additional metrics that we will check after the allele calls are made. But so far, for this particular strain, everything looks good. So that's kind of the same check that you would do for all the organisms, and we will post a sort of a quality metrics chart to get you acquainted with what values to be paying attention to and what numbers you should be looking at. And for the analysis certification that has just recently opened up to the laboratories, that's part of the analysis certification, so you'll be able to see that in the SOPs. But once you're satisfied with the quality of your strains and you're ready to export, all you have to do that is go up to File and then Transfer Current Selection. And what's happening is there's some settings that you set up beforehand, but is exporting these entries to specific folders that have been mapped to your organism-specific database. So I would expect if I went into my Salmonella database, I'd have one entry new to import, and so the same for the rest of these. So that is basically what there is to show about RefID and what it does. Is there any questions about that before we move on to the organism-specific database? Okay, well then we can move on to the organism-specific database. So I can now close this one, and we're going to work in E. coli. So I have an Escherichia database created, so I'm going to open that. And if everything went according to plan, we should have one new entry to import. So this is what the new Escherichia database will look like. This is a combined database with both PFGE and WGS data. We have a lot more experiment cards now, as you can tell. Escherichia is a big one because it now combines with the Shigella databases. So all these dots represent present experiments that are associated with the number up here. So if we have number 1 is the antibiotic experiment. If there is a green dot for that number 1, that means that experiment is present, that has been run. 3 would represent a de novo assembly, and so on. So you know you'd have sequence data in here. And then the rest of these are related to PFGE patterns. But we want to go ahead and see if the export works for importing data into the Escherichia database. So what I do now when I come into this database is I go back to my file menu and I choose the option for check for data transfer updates. And it tells me here that I will not be creating entries, but I will be updating an entry. So what could have happened in this case is maybe I ran sequencing on an isolate that I already had PFGE data for. So I'm just now linking the sequence data to that PFGE entry. So this is it's really important for you all to remember to use the same key names that you're using in PFGE that you're using for sequencing, so that all the data links to a single entry within your BioNumerics database. If there are any changes to that key name, even if it's a space or a dash versus underscore, it'll create a new entry, and then you'll have PFG data and sequence data, of course, linked to 2 different entries in the database that are supposed to be to the same patient. So in this case, we are updating an entry, and that's fine. So I'll choose OK. And it told me that it imported 1 entry successfully. So that's a good sign as well. So I choose OK. And then I can look down at the bottom of my screen and I can see that I do have 1 entry selected. So if you wanted to hit Control-T, that should bring that entry to the top of your BioNumerics window, and you can verify that, in fact, it did bring in the sequence data. You do have a de novo assembly associated with this entry that also looks like has PFGE data associated with it too. So at this point, you need to get allele calls. You want to know that you need to be able to compare it, of course, by the other sequences in your database. So you just go back to that cog to submit jobs to the calculation engine. And at this point, you're going to submit for your assembly-based allele calls, also assembly-free allele calls. You can also submit for genotyping. This will give you the serotype, pathotype, virulence, antibiotic resistance information associated with this particular strain. So all these 3 should be checked. De novo is not checked because we just did that in RefID. So that should be unchecked because it's already present. And then we choose OK. And we spin our wheels for a moment.
Hi, Stephen. This is Alice in Colorado, and maybe while the wheels are spinning, I'll ask a question.
Sure, perfect.
Can you, and maybe others are more familiar or aware than I am, but can you clarify when most agencies will get access to this version?
Yeah, so we are currently deploying out the software to the states, but right now it's only for PFGE use because we're still finalizing some of the tools. As you can see, there are a couple little buggy things that we are in the process of working out, which should be worked out very soon. We're also having to reanalyze all the sequence data that we have accumulated over the last couple years from your sequences. And once that is all finished, then we can open it all up to the rest of you. So we're hoping that by January, sometime in the middle of January, you'll have access to the whole genome sequencing component, but you should all be converted, hopefully, to this version of BioNumerics for at least use for PFGE by the beginning of the year. Maybe at some point in early January. We're aiming for our January 15th, quote unquote, end of PFGE date. Okay, well, it was always good to be safe than sorry, so I did prepare this as well. So what would happen though is we would see that those entries would upload just as it did in reference ID to the calculation engine the same way. So you'd end up with this box with those entries waiting queued for all those jobs to finish. And then what you should see is a finished result that looks like this. And then you select those entries and you pull back those results. Okay, we're almost there here.
Stephen, if that takes a sec, we've got a good question in the chat box, which is,
oh, sure.
What is the advantage of having a local reference versus using NCBI?
Well, the local reference is a way to just basically import your sequence data as it comes off of the sequencer. So you can get answers within just a couple hours. If you upload it to NCBI, then it takes some while for them to post it and you get those results. It also does additional quality checks that we have standardized here. And it's just a link in order to basically make sure you have good quality data before it goes into your reference database. The biggest thing that we see happen a lot right now is a lot of laboratories will mix up species. For instance, I'll get something that's supposed to be associated with a Listeria strain, and it turns out to be a Salmonella, but I've analyzed it all through my Listeria database, and now I've created a bunch of allele calls or allele calls that aren't associated really to anything that it would match to. And then we figure it out through ANI later, what it was supposed to be. So this is a way to capture some of those mistakes before it gets into your local databases. And it does it pretty quick. Okay, so we've now imported the genotyping results and other results into the database for these two strains. So to show you kind of what that looks like, let's see, we have our WG-MLST, which is in 16. So if I clicked on the 16 dot, what you would see is all the loci associated with E. coli and then the allele calls associated with those loci. We also have experiment presence for plasmids, resistance, STX, and virulence for E. coli. So if we clicked on plasmids, we would see whether they were absent or present for these different characters. What is 21? 21 resistance, the same thing. Present or absence for those different genes. STX, this looks like it's STX1. It's an STX1 in EAE. And then 23 virulence, it shows you what's present and absent for that as well. And then it captures some of this information also in the database. So if you scroll over to some of those database fields, you will see that it determined that it's an STEC. That is 045H2, it's STX1A. And if we look at our traditional serotyping results, which we had in the database from what we got from our reference laboratory, this was already identified as an E. coli 045 with an undetermined H antigen. So in this case, luckily for us, we got the same results virtually with the genotyper. Okay. Now at this point, you would assess the quality of the strain. So again, you can go back to that quality experiment, number 15, and you can click on the green dot and go through the other quality metrics that might be captured. Some of the additional things we look for after allele calls are made is, for instance, the core percent. This number should be 95% or higher. So in this case, this strain was found to have the core percent of the genes identified at least 95%, so that's good. The second one, 98%. So these would pass our quality thresholds, at which point we would be ready to upload this data to the PulseNet National Database. So if we were to do that, we go to PulseNet, and then we do web logon. Of course, we would authenticate to the PulseNet firewall first, and then you would enter in the credentials that you would get upon your analysis certification. We should be logged on in just a minute. E. coli is now a pretty large database, so it takes just a minute to get connected.
It's good timing because we have another chat box question if you want
Perfect.
to take that one in the interim.
Good way to fill the time. Okay.
Right. What if the core percent is less than 95%? Would you need to resequence?
You would, yes, yes. And the reason that's important is that, that leads a lot to, of course, the nomenclature. The nomenclature is based on a 95% core presence at least. So if you ended up not identifying genes that should be associated amongst most of your genomes for Escherichia, then, or any organism really, then the integrity of the name may not be quite there. So we do expect to have that if the point is to have sequences that are passing quality, then they need to have at least the core at 95% found. And that's pretty doable. So that's, it's not a hard thing to achieve if everything went well with sequencing. Okay, so now we are connected to the server and we can now upload. So we have these two entries and we just go to a little blue arrow up here that says submit selected entries to the server. And it asks you what you want to upload. And because we've already uploaded PFGE data before, you can deselect the PFGE information. We're just interested in uploading the sequence information and the genotyping results. So we would just select those two things, as well as the quality and all the genotyping stuff and the WGMLST calls. It doesn't hurt to also upload the PFG. The problem though you might have here is if you already uploaded PFGE and you have confirmed PFGE patterns in the national database, you'll get an error message that says those patterns have already confirmed, and it kind of stops the upload. So that's the main reason. But if you're uploading PFG at the same time you're uploading sequencing data, then it wouldn't matter as much if you submitted them all at the same time. But anyway, we're going to go ahead and choose OK. Now it's preparing to upload these two strains to the national database. Are there any other questions? Well, I think we're almost at the end of it now. Let's see here. There we go. So upload completed, and it basically assigned us a WGS ID. So if we scroll over to our WGS ID field, you'll see that a WGS ID has been assigned for these two entries. At this point, it would be safe to upload those to NCBI if you're concerned about anonymizing your information there. So then you just go to this NCBI menu item and choose Submit to SRA. There you would enter in your login credentials for that, which will also be provided to you upon certification. And you'll have an option of templates to use. We have a non-clinical template and then a clinical template. The clinical template is, of course, for your human isolates, and for the non-clinical, that's your non-human isolates. There's a difference because a genome tracker requires more information to be released at the onset of submitting data to NCBI than CDC does for clinical entries. Some of those differences are, for instance, you would include for a food specimen the state in which it was found, the collection date. There might be one other piece of information that also gets added. Those are two things that we don't include with the initial upload to NCBI for clinical strains. So, but these are clinical, so we'll choose our clinical template, and then you just hit submit. And this is set up into a test environment, so it won't go, of course, but it would basically pull up another jobs overview window, just like you saw for the calculation engine, and it will show you the status of the upload to NCBI. Once that finishes, you can download the SAMN number as well as the SRR ID that's associated to your strain into your local database. Another thing you can do is request a WGS ID without having to upload. Well, you have to upload something to the national database, but say you have an old historical PFGE isolate that you want to go ahead and sequence, and for some reason or another, you want to request the WGS ID before the end of the process. So what you can do is select the entry in your database that you want to get a WGS ID for, and then go to your PulseNet menu item and choose Get WGS IDs. And this is basically looking into the server to see that what the if the entry exists, and if it finds it, it populates a WGS ID in your local database. So then you can move forward with your sequencing or whatever it was that you needed that ID earlier in the process as opposed to the end of the process. I know some people have developed workflows that have now utilized the WGS ID in different ways, and were a little bit unsure of how they felt about it now coming in at the end of the process. So you can get it at the beginning if that's something you need to do. Okay. So I'll try to quickly go over some of the analysis stuff. So now at this point, you basically imported things into reference ID, you've speciated it, you've submitted it for allele calls and your genotyping results. Now you want to do the fun stuff, and that's the surveillance. So what you can do, one way to do it, there's many different ways, of course, but anything that has a WGMLST experiment associated with it has sequence data. So you can hover over 16, which is representative of that in this database. I can right-click it, and I can do select entries with experiment. That selects everything in the database that has a WGMLST experiment, which are the allele calls, and I can pull those up into a comparison. You can then create a dendrogram based on those allele calls. So we'll click the eyeball next to WGMLST in the comparison view, which will pull up the loci and the alleles associated with those loci for these isolates. And once that's done, you can click this quick cluster button. It's the cluster looking button with a lightning bolt through it. We'll see how quick it is. So this is going to create basically a UPGMA tree based on the allele calls here.
We have another quick question while it's spinning.
Sure.
Animal, non-human, clinical should be uploaded as clinical or non-clinical. So animal would be?
Animal would be with the non-human.
Okay.
Yeah, good question. Okay, so now we have our dendrogram, and you're looking at sequence data here. And unlike PFG, where you have patterns that you can look at and see, okay, these are indistinguishable and these aren't, it's a little bit more difficult to determine when looking at sequence data. At what branch is too much branching versus, is a flat line what I'm looking for? Am I looking for something that's kind of a flat line, like, is something like this here a cluster, or is this the cluster, or is it a cluster at all? I don't really know. Well, one of the tools we have is a way to label your dendrogram to find out. So you are looking for more of your flatter lines. That's your indication that you might actually have a cluster based on sequencing data. Something that branches out on this level is definitely probably more branching than we would expect to see in a cluster. But one way you can do this is you can go find your, what you think might be a cluster, and you can right-click on the node associated with those entries, and then you can do label tree node with within branch distance. And what this does is it puts a median, minimum, and maximum allele difference of these three isolates in that branch. So this looks like it's two allele differences, which I would consider pretty close. Well, then you might be curious, well, what are the two below it? Are these guys also related? You can go click on where that node connects all these guys, do the same thing, and find out that we're now up to 68 allele differences. So we know the bottom number, or the bottom one, isn't related. The median's 46, so it's likely this one isn't either, but these three might indicate you have a cluster. If you wanted to see a pairwise comparison of these allele differences, you can do that. You can select, say, all 5 of these if you want to see exactly how they compare to one another, and then go to File, Export, and Export Differences. You'll get an Excel spreadsheet that pops open over here. That gives you a pairwise comparison of those 5 strains to each other, so that you can see, okay, this guy relates to this one at 2 alleles different, but this one's 51, and it does it for all those in the selection. Another neat thing you can do, let me see if I can find an example of it. Might be an example like this. This is not a good example, but it sort of illustrates the point I'm going to try to make, is you have 3 groups, which say these were looked closer related, but you have 3 groups, and you want to know, okay, well, how does this group relate to this group? Well, that's easy. You can click on this node, and then in this case, you can do label tree with between sub-branch distances, and it will give you the difference between this guy and this guy. But then you might want to know, well, how does group 3 compare to group 1? You can't really do that with this labeler, because if you can click this node, but it's going to include all of these together, not just this one compared to this one. So we have worked with Applied Math to develop a way to do that, and that's through using grouping. So what I can do is I can select everything associated with this group, and then I can go to groups, create new group from that selection, and call it, group 1 maybe, and then do the same thing with this group down here. Oops, let's not cut that out. Let's give it a group and call it group 2. Oops, I should have left. Okay, so I have group 1 and group 2. I could select those, and I can go back to File, and this time do Export Group Differences. And now it tells me that from group 1 to group 2, this is what the minimum, or median, minimum, maximum is between those groups of strains. We do a lot of this with HQ SNP trees, where you have different groups that fall at different places of the tree, and some groups that fall at the top of the tree are not actually that distantly related from groups that are up at the bottom of the tree, but the way the topology works, it just kind of moves them around in different ways. The same thing can happen here. So we were working with Applied Maths to find a way to be able to compare those closely related groups to each other without, having to just rely on the whole group together, but, doing exactly what I just showed. So that's sort of a new feature that has just come into play here. But yeah, this is kind of how you detect clusters right now in, I guess, Escherichia. We are working to develop a nomenclature, which would make this a lot easier. We have developed this nomenclature already for Listeria. So to go back to the PowerPoint, I'll talk a little bit about that to wrap this up. We have Listeria Nomenclature that's been developed now since the beginning of the year. We've made a modification to it in the middle of the year, which now means that it has 6 numbers as opposed to 5. The nomenclature tells you the organism, the version of which the nomenclature was used to build these names, and then it starts at a number that represents matches in the database up to about 71 allele differences. And then if it has a match on a second number, then it has those match each other within about 51 alleles, going on down to about 0 allele differences based on the core genome. We find for Listeria cluster detection using core genome MLST, the sweet spot being around 7 allele differences or less. So when we're doing cluster detection there, we're basically looking for matches up to the 5th number. We will consider things, of course, as we get more information, or some things that might be closer on the threshold of 7, or maybe it's 8 alleles different, so it doesn't get the 5th number, but it does match at the 4 numbers. So we could go back and look at those as well if we match everything on the 4 numbers. But we typically, for just cluster detection like we did with PFG, looking for indistinguishable matches to start off an investigation, we kind of identify things that match on the 5th, matching 5 digits. So to quickly illustrate that, I'll have to pull up my Listeria database, because this is the only place, like I said, it exists currently. And this is what the nomenclature looks like in BioNumerics. And we expect that once this is implemented for all organisms, this will become an automated process. So you'll upload your entries to the national database, and within less than a day, you should have a name that you are able to download back into your local database. And like I said, the numbers look like this. And to do cluster detection, you're going to have some functionality that we've built for Listeria that's going to be similar for the other organisms as well. Here it's an option called cluster search. And all we have to do is click that, and we can click Find Clusters. And it pulls me up all the found clusters. So what this is built on by default is matching on the allele code up to the 5th digit. That's what these numbers of 5 represent. It includes anything that's at least a cluster size of 3. And it's looking only at the past 120 days. These are all just the default options for our national surveillance, but this can be manipulated any which way you want it to be. When it's in your local laboratory, you can change it to a cluster size of 2. You might be interested in everything that matches up to, the 4 numbers instead of the 5. And you might, choose a longer time frame to go back, or a shorter period of time to go back. You can also choose to include non-human sources. So if there's, environmental or food samples in your database that may match this clinical strain, you can do that. You can also choose to include all historical matches. So, you're basically ignoring the 120 days, and you're just saying, give me everything that matches that allele code if 3 or more has been seen. But what I like to do is, start small and then kind of widen it out if I need to. So I can go up to my 5 numbers. I can leave it at 3, 120 days. Anytime you make a change to these settings, you just hit recalculate, and that'll readjust what clusters do you have. And if I wanted to look at some of these, I can click, for instance, this cluster, and I can choose OK. And it's going to pull up a comparison with those entries. And then I can create my tree. And if I wanted to see, of course, how related these are, I can just right-click on that tree and then label node, and it's these guys actually, by whole genome MLST, aren't as related as they look like they were by core genome MLST. So again, remember the name is based on core genome MLST, but when you create a tree, unless you change the view to show only core, it will create a WGMLST tree. So to do that, all you have to do is click on this all characters and choose your core scheme as opposed to all loci, which is your whole genome, and then recreate the tree and you'll get different numbers. So let me go back to the E. coli database to illustrate one other thing. So if we have sequences in the database that we were curious about having matches to, you can search on the core scheme to do that. So say you found a cluster and you want to know if there's any historical things, or say you just match everything against your database every single time you bring in something new. All you have to do is select those entries, and then you go to your WGS Tools menu item and choose Fast Character Matching. And this allows me to choose the experiment type I'm matching against. So in this case, it's the WGMLST experiment type, but I want to look at the core scheme. You can match it on any of the schemes. You can do it on any of the MLST schemes, plasmid schemes, chromosome-associated scheme. The whole genome MLST would be, again, the all characters or all loci, but we're going to do core. If you're wanting to only pull in quality strains, you can put in some quality filtering by only finding matches that match up to 95% core found, or 95% or higher. If you want to see everything regardless if the quality is good or bad, you can just uncheck that. If you want to filter it by a date range or a database field, we think this might be helpful, especially for like Salmonella when you're searching what could be a very large database. If you say, okay, I have a Salmonella enteritidis, I only want to compare these strains to Salmonella enteritidis, you can select your serotype field as the field to search in so that it doesn't search your entire database for matches. That could take a little bit longer. Then you determine your allele difference threshold that you want to look at. So if you're looking at, I want to see everything within 10 allelic differences, you can just set your primary and secondary matches at 10 and then choose OK. And that's going to take these 5 strains and it's going to match it against the database. It told me that it did find matches, but they were of low quality, and that's OK. And there may have been a problem with one of those, so let's try this one. Well, apparently I need to unpack 2 values. I'm not sure what that means. But
Stephen, would you mind if we pause just for a sec to answer if we have a few?
Yeah, no, that's fine. That's about all I have to show at this point, so we can ask questions.
Let's start with one in the chat box. How far can we go in BioNumerics for those species that had a star by them in the ANI PowerPoint slide?
Oh, sorry, I was on mute. So there you can identify them in ANI. I would not import them into your organism-specific database because once you run allele calls on those, you're going to maybe not get any or just very few for a lot of those species. So if you're pulling it in there to look at the WGMLST results, you're not going to have good results. If you want to pull it in there to do a SNP analysis, then you can do that.
All right, so the next question is, what is the scalability of this method? And I think this is referring to the nomenclature scheme that you showed. As in current example, you showed only 5 samples. What number of samples can be analyzed efficiently?
So are we talking about cluster detection or are we just talking about naming? I mean, you can basically, it runs it pretty efficiently in terms of cluster detection. It basically scans the whole database and pulls up clusters based on your defined thresholds in that settings view. In terms of the names growing, we base the names on a tree that's been built with everything we have already in existence for the nomenclature. I mean, so for all the stuff that we've sequenced over the years. And then, I mean, it seems to be pretty scalable. I don't know if Heather, you have more to add about how the nomenclature is built on the tree. I don't know if Heather can talk. I don't know.
Can you hear me?
Everyone should be able to talk.
Okay.
You might be doubly muted. Sometimes it's your phone and computer. You both have to unmute both sometimes.
Can you hear me?
Yeah, I can hear you now.
Oh, good. I was trying to unmute and then I forgot what the question was. Sorry, what was it again?
I think it was referring to the scalability of the nomenclature. So I was trying to answer how we develop the nomenclature based on, thousands of strains. And, in terms of the scalability, it seems pretty robust, but I don't know if you had any more to add to that.
No, we developed the nomenclature specifically for the different organisms and developed the thresholds to be able to adequately identify, say, an S-tech outbreak versus a Listeria outbreak. So we have multiple points along the multiple numbers to reflect the amount of diversity we expect to possibly detect in an outbreak. So it should be fairly scalable, and it's pretty robust it's pretty straightforward to name new isolates and put them in our overall nomenclature scheme.
I think we were also thinking about Salmonella, specifically the examples are for Listeria and E. coli, where the number of isolates is a lot smaller. So when you think about thousands of